Hello, I'm Professor Charles Knight at Cal Poly in San Luis Obispo, California. I work in the Bailey College of Science and Mathematics and in the Department of Biological Sciences. And this is what I'm calling my 10 lectures on evolution, partly for my Bio 414 class, Evolution. It's an upper division class on evolutionary biology, but also for you out there on the internet. This is the target paper for today. The title is Convergent Evolution of Plant Prickles by Repeated Gene Co-option Over Deep Time. It was published in Science on my birthday, August 2nd, 19 or my birthday is August 2nd, 1972, but it was published in 2024 and it was by a whole bunch of people, and I just have a chance to highlight a few of them. Of course, I start with the last author and the first author, Zachary Lipman and James Shatterley. But I just want to say up front that there's a whole bunch of people in this worldwide collaboration that eventually produced this science. I say eventually because there was a bunch of preliminary work that went into it, I assume. Well, I know, but also independent small studies, probably multiple PhD dissertations were involved in this paper, I would assume. But let me talk a little bit about Zachary and James. Um, Zachary Lipman is a professor and a Howard Hughes Institute investigator who works now at the Watson School of Biological Sciences at Cold Spring Harbor. And that Watson is the James Watson who used to lead that effort there at Cold Spring Harbor for a long time after he discovered the structure of DNA, which is a pretty big accomplishment. Zachary Lipman, from what I could glean from the internet, I've never met you, Zachary, but I hope to someday, um, grew up working on a pumpkin farm. And I would imagine that that easily would have engendered a lot of interest in variation in plants. Pumpkins vary by size by so much. There's contests all over the United States in October for the largest pumpkin. And even Zachary, it says, used to engage in those contests, and you could win money doing that. It's a pretty competitive sport in some places. I just wanted to also point out that pumpkins vary in size, but they also vary in lots of other traits, including seed size, a trait that's near and dear to me, but so many other ones too. Some of them very discrete, but many of them quite quantitative, as we'll see soon. I'd also like to, well, also Zachary went to Cornell as an undergraduate and then got his PhD at Cold Spring Harbor Lab, and now is a full professor and leads his own lab there at Cold Spring Harbor. But James or Jack Satterley was a postdoc with Zachary Lipman when this work was produced. And so he's actually James or Jack is just starting his own lab now in the Department of Botany at the University of Wisconsin-Madison. Uh, Again, before that, he was working with Zachary at Cold Spring Harbor, and he's just recently moved to the University of Wisconsin-Madison. Um, Jack started his academic career at Macalester College, which is a small liberal arts school in St. Paul, Minnesota. I believe it's in Minnesota. It's right at the confluence of a number of different states there in St. Paul. But he's had quite a career so far, and I look forward to continuing to follow Jack's work. I just wanted to mention again that there was a worldwide team of investigators. So many, it's too long to go into here at the start of this paper, but I just encourage my students to pay attention to some of the names because these people are part of a small community, especially a small community of investigators studying quantitative traits in plants. I know Zachary worked with Stephen Tanksley and Rob Martinson before he ended up at Cold Spring Harbor. And obviously, Jack was working with Zachary Lipman. It's a small group of people that are doing this work, and uh, I encourage my students to start to learn the names of these people because they'll continue to see them throughout the years. Uh, I'm emphasizing here that there were a lot of resources that made this presentation comparatively easy. And one of them was that science produced, I'm sure that the authors produced really, but science encouraged the authors to produce a research article summary. I'm not quite sure what that process is, whether the Zachary and Jack were like, we could easily do a research article summary, what we sometimes call a graphical abstract, and what I'll refer to here as a graphical abstract. But there's a really good one at the beginning of this paper, and then right after the graphical abstract, it goes straight into the main publication. Um, and so I encourage my students who are looking for papers to do a presentation like this. If there is a graphical abstract, it may be very useful, or it may not, it depends. And I'm going to use this graphical abstract off and on during this presentation. Also, this presentation was made easy because Elizabeth Kellogg, or AKA Toby Kellogg, also wrote a perspectives piece in Science in the same issue about this paper. And these perspective pieces, I think they're called perspectives in Science, maybe it's News and Views, but this perspective piece is a small one-page paper that kind of places the results in a larger context for a broader audience. But Toby made a great perspective piece that highlighted some of the issues, some of the significance of the findings of this paper, and it really made it a lot easier for me to construct this presentation. I just wanted to point out that Elizabeth Kellogg is now at the Danforth Center at the Missouri Botanical Garden. And she has this, she's also a member of the National Academy of Sciences. And on her website, she has this great quote, which I can't help but share now. And that just says that it's been said that there are only two kinds of organisms in the world: plants and plant parasites. All life depends on plants. And I love that quote. And as soon as I tell it to somebody, they say, well, but what about herbivores? And, those aren't parasites. And I'm like, no, they actually are parasites, I would say. So that's a great quote. Thanks for that, Toby. But here we go. I'm also going to foreshadow a little bit to our target paper number 5, which will follow, I think, next week. Um, and this paper is a little bit older. I think it's from 2018. But the title of this paper is Predictable Convergence in Hemoglobin Function Has Unpredictable Molecular Underpinnings. And the reason that I'm showing you this here now is that the conclusion of this paper is very relevant to the conclusion of that paper. And this whole concept area of trying to predict the molecular underpinning of convergent traits has a history that I want to present right now. In that paper, in this target paper number 5, they say, uh, in a highly influential paper in biophysical mechanisms of protein evolution, Pruitt's predicted that adaptive changes in functional properties of vertebrate hemoglobin are typically attributable to a few replacements at key positions. And there's a picture of Max Pruitt's right there. He won the Nobel Prize in 1962 for this work on hemoglobin. And next week, we're going to discuss this paper. But this is a big deal because a lot of people didn't think that that would be the case, that there would be predictable or even mutations in the same area. And I just want to give you the first couple sentences of that paper by Max Pruitt. This is the paper in 1983. It was actually in the first issue, the very first paper in the journal Molecular Biology and Evolution by Max Pruitt in 1983. He said that species adaptations at the molecular level is a virgin field. I'm laughing because it seems like my entire class in evolutionary biology deals with the molecular level of evolution and adaptation. He goes on to say that Lewontin in 1979 complained that it has proved remarkably difficult to get compelling evidence for changes in enzymes brought about by selection, not to speak of adaptive changes. Such evidence has recently been gathered for hemoglobins whose response to different chemical stimuli varies widely in vertebrates living in different environments. So Pruitt is just kind of setting the stage here in those sentences for reviewing his work on hemoglobin evolution, which is a canon within the field of molecular evolution. It's not surprising that this journal, in its very first issue, in its very first paper, would ask Max Pruitz to write a synopsis of his work. But we're going to go back to this at the end of this paper. Remember, class, that the search for homologous genes is quite futile except in very close relatives. That's what Ernst Mayr said in 1963 in his book Animal Species and Evolution. I took a deep breath. My students know that there's a lot that is packed into those deep breaths because Ernst Mayr was just wrong, really just wrong in many ways. And remember his next quote that there are many roads that lead to Rome, and that is true in evolution as in daily affairs. And again, you could think about this in several different ways, but this particular quote I found is not directly attributable to Ernst Mayr, at least by what I can find on the internet right now. Our class was introduced to these quotes from Endless Form's Most Beautiful. And really, I just wanted you to think again about this topic, about whether convergent evolution is attributable to a few genes or a few direct causes, mutations at specific locations, or if there's mutations all over the genome. And we're going to get to the answer that it's a combination of both, but in this case, they're mutations in the same gene, the log gene or the lonely guy gene. So I'll get back to our target paper today, which is Convergent Evolution of Plant Prickles by Repeated Gene Co-option over Deep Time. And as I normally do, I'm going to give away, I'm going to tell you my I think it's 6 take-home points from this paper. First is that prickles have convergently evolved and been lost many times. And in some cases, we're going to describe the convergence as loss of prickles, not the gain of prickles, but the loss of prickles is a convergent trait. The extent to which convergent phenotypes rely on shared molecular traits, that is, genes or regulatory mutations, is hotly debated, as I just highlighted in the last slides. Here, in this paper, the authors show that convergent losses of prickles share similar but different mutations in a cytokinin gene called lonely guy. QTL analysis and fine-scale mapping were used to find the genes, and I'm going to go into a little bit of detail on those two techniques since this is a class in evolutionary biology. So we'll study a little bit about how those are done. And CRISPR was used to re-engineer pricklelessness in prickled wild relatives. And this is really a cool advance that has been made possible by CRISPR technology. And it's kind of just seals the deal that they've found mutations that cause pricklelessness. And lastly, the authors picked a simple, non-pleiotropic trait, and it's not always this easy, is what I say to my students. This is a fairly discrete trait. For many quantitative traits, by definition, they're continuously varying, and it could be much harder to do anything like CRISPR to uncover or re-engineer the phenotype. Possible, but just want to highlight some reality here. They were smart to pick a fairly simple trait. All right, so as I normally do, I read the abstract because the authors put a lot of thought into these words. I think it behooves us to start there. So I'll read the abstract slowly and then go into some confusing or definitions of words that I think are important. So, Convergent Evolution of Plant Prickles by Repeated Gene Co-option over Deep Time is the title. An enduring question in evolutionary biology concerns the degree to which episodes of convergent trait evolution depend on the same genetic programs, particularly over long timescales. In this work, we genetically dissected repeated origins and losses of prickles, which are sharp epidermal projections that convergently evolved in numerous plant lineages. Mutations in a cytokinin hormone biosynthetic gene caused at least 16 independent losses of prickles in eggplants and wild relatives in the genus Solanium. Homologs underlie prickle formation across angiosperms that collectively diverged more than 150 million years ago, including rice and roses. By developing new selenium genetic systems, we leveraged this discovery to eliminate prickles in wild species and indigenously foraged berry. Our findings implicate a shared hormone activation genetic program underlying evolutionarily widespread and recurrent instances of plant morphological innovation. Pretty dense, a lot of words there, but let me dissect a few of them and get us started here. I circled these here and we'll continue on. Some of the words are not just words, but maybe concepts like a shared hormone activation genetic program. That's a pretty large phrase, and widespread and recurrent instances of plant morphological innovation. Those are kind of just large phrases there, but let me go into some specific ones in what I call the glossary notes. I put this in class when students should probably take some notes and actually write things down. So I kind of slow it down to a pace where students could actually take a note here. Prickles, for example, are sharp epidermal projections, and these are not modified leaves or stems or branches. They actually are just outgrowths of the epidermal cells. Convergent evolution, just to keep it simple to begin with, is the evolution of similar phenotypes from different genetic backgrounds. I think we're all familiar with that. But here we see 16 independent losses of prickles. And here we see a convergence in losses instead of gains. I'm still trying to get your perspective to switch that a loss could be a convergence just as well as a gain of prickles. Homologs are biological features that are similar or identical because they are descended from a common ancestor, and homologs can be genes, chromosomes, or other features. Our class has been studying orthologs and paralogs. Those are two different types of homologs, which are similarities from a common ancestor. In the case of orthologs, they're in different species. Paralogs are in the same species. Does the convergence come from a shared genetic program? In this case, the answer is yes and no. It's the same gene, but different mutations. So it's a little bit shades of gray here, but remarkably similar, I would say. The same areas of mutations in the same gene. And this is part of a shared hormone activation pattern. I'm pointing this out because I'm reading another book right now called Evolution Evolving by a number of prominent scientists, including Scott Gilbert and Marcus Feldman, who are emphasizing that a lot of developmental biology involves gene regulatory networks. And here we have a gene regulatory network that's involved in hormone activation. And we know that cytokinin genes are important in this, and there's been a convergence here. There's different mutations in this particular gene called LOG that are associated with this convergence. And I'm smiling now because LOG, the name of the gene, is called lonely guy. And I'm going to show you why it's called that. It's kind of a cool little study. So I got to take another step back because this is a class. We go back to the textbook that the students aren't reading. They're only just using it as backup information. This is what I call a textbook tie-in from chapter 9 of Freeman and Heron. And that chapter is on evolution at multiple loci and quantitative genetics. And this may seem like a little bit of an aside because the trait that they studied was really just fairly discrete. But they get to the location of that gene using what's called fine scale mapping, which is very analogous to QTL analysis of quantitative traits. So I feel like it's important to start with QTL or quantitative trait loci analyses. And that discussion always starts with what is a quantitative trait. And quantitative traits are those that are continuously varying. On the previous slide, I forgot to mention those were humans and they varied in size. The black ones were the males, the white ones were the females, and in both groups there was a continuously varying distribution of heights. And so we could say short to tall is a quantitative trait in humans. In pumpkins, it would be small to large. That's a quantitative trait. And finding the genetic basis of these quantitative traits is the science of quantitative genetics. And I encourage my students to tell their parents that they're studying quantitative genetics because I think their parents will be proud. It sounds really complicated. It's actually not so bad if you stick with me. So scientists wondered how well Mendelian genetics would describe the heritability of quantitative traits. It seemed like this was going to really blow up the field. Mendel's laws were discovered, in the 1930s, 40s, 50s, something like that, or rediscovered, and really found the genetic basis of traits in general, that they could be these particulate pieces that were inherited. The Mendelian genetics really caused a revolution and led to the modern synthesis of evolutionary biology. But then these quantitative traits were observed that continuously vary. And actually, before Mendel's laws were rediscovered, Edward East did some very relevant experiments that showed that quantitative traits do behave in a Mendelian fashion. It's just that there are multiple loci. And he studied corolla length in Nicotiana longiflora, a tobacco plant. And what he did is he crossed, I'm going to lead through 3 different experiments, or, well, they're kind of experiments here, or at least beginning a thought experiment. We look first at a trait that's controlled by one locus, little a, and a big A. If you have parents, a homozygote short, little a, and a homozygote tall, big A, and these are true breeding, they would continually produce those phenotypes if you bred them together, well, within those classes, short or tall. But if you cross them and breed what's called an F1, or the first filial generation, you get complete heterozygotes. And in this hypothetical and actually experimental situation, you get a completely intermediate phenotype that's medium height. And if you breed those F1s together, you get what's called an F2, second filial generation. And in that generation, you have 3 phenotypes: short, medium, and tall. It's a very discrete distribution: short, medium, or tall. That's all you get. It's not really a quantitative trait. When we expand the model to 2 loci, where the short parent is little a, little b, and the tall parent is big A, big B, breed those together to produce an F1. The genotype is completely heterozygote, little a, little b, big B. And the distribution of traits now in the F2, if you take that all the way down to the F2, is more continuously varying, closer. And if we expand that out to a 6 loci model, I'm not going to say all the letters, but you get it at the F1, get a completely heterozygote across 6 loci. And the phenotypic distribution in the F2, once you breed those F1s together, is what very much resembles a continuously varying quantitative trait. So the conclusion of Edward East's experiments was that, yes, quantitative traits do behave in a Mendelian fashion. It's just that there's lots of loci. But he also made 2 predictions. And one of them is that for quantitative traits, F2 plants will only very rarely exactly resemble their parents. And secondly, that within a few generations of selective breeding of the F2 plants, you could restore those parental phenotypes. So if we test the first quantitative traits, and these are the real data from Edward East's experiment, you have the parental genotype or distribution of traits: short corolla length at the beginning on the left, and long corolla length for a parent on the right. There's variation within those parents, but in general, it's a non-overlapping, completely different phenotype of those parents. You breed them together, make an F1. Those are the hybrids or the first generation children of those parents. and you get an intermediate phenotype, a little bit skewing short, but not so much. And then breed those together, produces an F2. And what you can see is that in the F2, almost none of the individuals actually were exactly the same as the parental individuals. At least maybe on the tall side, there might be one that overlaps with the distribution of tall, but on the short side, I don't think there's any that overlap with the distribution of the small parent. So Edward East's prediction was true, but of course he made this prediction after he observed this. Uh, but then he tried to test his second prediction, and that was restoring the parental phenotypes after selective breeding, or just pulling out the tall ones, breeding those together, pulling out the short ones, breeding those together. And sure enough, he gets short and long corolla length Nicotiana that did very much overlap with the parents. So you could restore these parental phenotypes, and that was after only 5 generations of selective breeding. So this is just a background of quantitative genetics, and that method of breeding is going to be important. The question here, though, even for Edward East, but I don't even know if he had the vocabulary or theoretical basis to wonder this, is what are the genes or alleles that are being selected for? What is it that you can select for? Even in 1916, they were really in the dark ages of genetics compared to what we know now. So I'm going to go with some more glossary notes here. Quantitative traits are traits that are continuously varying. Discrete traits are traits that are categorical. They're either there or not. And prickles, you could say, is fairly categorical, but we'll see there's some variation. F1 are the children of the parents, or the first generation, called the first filial generation. F2 are the progeny of the F1, which are inbred children of the F1. So that has its own problems, but it's very useful for genetics. And QTL analysis is an experimental or statistical technique that's useful for finding the genetic basis of quantitative traits. And I'm going to say, really, it's just an association study. It's fairly simple. I want everybody to remember that recombination takes place at meiosis 1, prophase 1 of meiosis 1, about once per chromosome arm. These chromosomes cross over and exchange genetic information. And that creates new combinations of alleles, and that will generate lots of variation in populations. And in the F2 population, you get lots of variation because of crossing over. It's not new mutations. Crossing over is a large-scale mutation, but it's not a point mutation that causes the variation in the F2. It's this recombination, which is a much more likely route to generating the variation, as well as independent assortment. Remember also that some instances can involve unequal crossing over, and the only reason I emphasize that here is that our previous papers on amylase evolution involved gene duplication, also on alcohol dehydrogenase that also involved gene duplication. So I just want to help the students remember that unequal crossing over is an important route towards variation in gene copy number. But in this case, we're really just focusing on recombination in general, producing a quantitative trait distribution, lots of variation in alleles, not necessarily unequal crossing over. This was the figure that we were using for unequal crossing over. Still looking for a good video animation of both recombination and unequal crossing over. Neither of those are very well represented in the field, in the canon of what you can see on the internet. I'd like to see a better one out there at some point. I can picture it in my mind very well, but still haven't found a good one to share with my students. So I want to tell you or give you a map of how to do a QTL or fine scale mapping study. I took a deep breath because, similar to the limitation of not finding a good internet resource for unequal crossing over, the same is true for QTL. QTL presentations on the internet go very dry, very quick. And I'm going to unfortunately have to do the same thing. But I want you to stick with me because it's not that complicated. A QTL study first starts with identifying extreme phenotypes in a model population, a population that's easy to breed, easy to take care of, generates lots of seed, doesn't require a lot of attention, has a short generation time, probably these days has a sequenced genome, easy to do genetics with, or maybe even CRISPR, as we'll see. Not a requirement for QTL at all, but knowing the genetics of the system really helps. So we're going to identify extreme phenotypes. We'll just use Edward East's example with short and long corolla lengths here. We could have picked pumpkin size, small versus large. I'm sure somebody's done that. If not, Zachary Lipman. I didn't look that up, but I'm just trying to give you a map of how to do QTL here, or instruction set. The second step is where it starts to get a little complicated, but it's not. You identify lots of polymorphic SNPs throughout the entire genome. This would be more targeted in fine scale mapping, but for a general QTL analysis, you have no idea what the genetic basis of the trait is. You got to find markers throughout the entire genome that are present in the short, but not present in the tall, if we use that short versus tall comparison at the top here. Polymorphic means it's there or not. So a polymorphic SNP is there in one parental phenotype, but not there in the other parental phenotype. So these are clues or breadcrumbs that say this part of the chromosome might be associated with tall. The second thing that you would do is produce an F2 population, taking advantage of the fact that recombination is always going to happen during sexual reproduction. That generates lots of variation, lots of recombination takes place. It breaks up combinations of alleles and genes that are normally together, really just distributes the genetic variation throughout the entire genome. But you have a trick because you've got these polymorphic SNPs in the parental generation. Now you're just asking the question, where are those polymorphic SNPs that are associated with the phenotype that you're interested in? So the fourth thing you're going to do is score the F2 population for the phenotype. In our case, in this paper, are the prickles there or not? In the case of the Nicotiana, you'll just measure the corolla length. How tall is it, or how long is it? I emphasize to my students that high throughput phenotyping is a good phrase to put on their CV. If you put that on your CV and send it off to Zachary or Jack Shatterley, I think their eyes will perk up. They understand the importance of high throughput phenotyping. Something easy to score is better, and finding a better way to score it is even better. Shows some innovation. So then you have to do the scoring of the presence or absence of the polymorphic SNPs, and this requires a lot of bench work. At least it did when I did it. 20 PCR machines, lots of gels, multi-channel pipettes. I suppose there's probably ways to do it faster now, but those technologies have passed me by a little bit. But it's still a lot of work to do on the bench, a lot of individual PCR reactions. My students would probably say, well, why don't you just sequence the whole genome? And it's like, well, you could, but then you get too much information, I think. But I wonder if the QTLs are scored by sequencing now. And back in the day, we did PCR reactions for each one of the SNPs, which involves a lot of PCR reactions. And then you simply statistically associate the SNPs, the presence and absence of those SNPs, with the phenotype. For example, in the long phenotype, which SNPs are there? And that gives you some breadcrumbs saying, well, the gene for long corolla might be in this region of the genome. Same is true for prickles. Prickles are here. These SNPs might be associated with the presence of prickles. The SNP position tells you the locus of the genes or regulatory mutations I just had to throw in there that are controlling the phenotype. So this is how you do it, how you get closer to the gene. Everybody wants to know the gene for, and this is a way to get closer to it. But remember, usually it involves more than one locus, and each locus only contributes a small amount. And so in the end, what we're discovering is the genetic architecture of the phenotype, to use a phrase that I like. So that's the goal and the output of a QTL analysis. If you're with me, hopefully you can see it in your brain that really what we're doing is correlating the presence or absence of SNPs with a phenotype. And those locations that have the SNP and also have the phenotype are more likely to have the genes that contribute to that phenotype. Then it may be required to do a more fine-scaled mapping, which is kind of the same process where you cross more, but you just have a better idea of the regions that you want to study and probably find more SNPs in that region that are associated with the phenotype. All right, now we're kind of getting back towards our story and also like bringing in some of the information from the graphical abstract and also from Toby Kellogg's perspective piece. There have been 28 independent origins of prickles. In plants in general, there were lots of different numbers, like 31 losses, 28 origins. I forget what exact group we're talking about, but the point at the beginning here is that prickles have formed many times and they've been lost many times in plants in general. I think that's all you really need to think about at this point. But remember that spines are different than prickles, and thorns are even, well, actually modified are prickles, but spines are modified leaves, and those are different than prickles. I'm showing you some spines on a cactus, which are actually leaves. It's hard, you can see well, anyway, that's what they're morphologically derived from. Prickles are just outward extensions of the epidermis. So there's been repeated evolution of prickles, and this is a comparative analysis. What's not even an analysis, it's just trait mapping on the phylogeny of vascular plants and angiosperms and eudiocots. And you see different groups, and the number in parentheses is the number of times that prickles independently evolved within those groups. So in roses, they evolved 9 times. In Asteraceae, they evolved 9 times. Monocots 6 times. But even going all the way back to cycads, prickles evolved. So lots of independent origins of prickles across the tree of plants. Here, just because we're doing it, looking at prickles, it's beautiful. You see lots of variation. It could be one of these books from Kew Royal Botanical Gardens. I have the one on seeds, fruits, and pollen. There could be one just on prickles. If we look at these photos, which are great, rose bushes, they have the thorns there, which are actually not prickle. They're actually not thorns. They're prickles. We see on Ceiba speciosa, that South American tree that's planted on campus here, prickles, large prickles on the trunk of the tree. And those prickles that are on the leaves of Ceiba speciosa are also prickles, outgrowths of the epidermis. I should have included a picture here. I'm surprised they didn't, because the picture of Ceiba speciosa leaves is crazy, and you understand that would have been a great adaptation to avoid predation by any animals in the Pleistocene megafauna. We see lots of prickles, and I'll show you one more image. Different datura, even Dipsacus has lots of prickles on their fruits and also on different structures. Not much to say here quantitatively, just that prickles have evolved many times. But we get back to that same question. What are the genes or alleles that are being selected for that lead to these prickles? What is the genetic basis, which seems to be a big focus of my interest in evolutionary biology? So we're asking this question: do shared genetic programs underlie these convergent traits? this convergent trait of prickle formation? Is it a shared genetic program or a shared gene regulatory network, or is it mutations in the same gene, in the same intron, in the same regulatory region? And is that answer true across increasing evolutionary distance? We really had no idea until this paper came out. Had a pretty good idea. Sorry, just talking a bit. So over increasing evolutionary distance, does this shared genetic program hold up? We'll see. So this is a figure from the paper. This is figure 1B. I'm just going to get a drink here. It's complicated enough. I thought you could take a minute to take a look at this. So this is a circular phylogeny of just one small group of plants. Small, lots of different species here, but notice they're all S. This is Solanium. These are the prickly solaniums, or that is solaniums where prickles are pretty common. Basically, all the species that don't have names are species that have prickles. The ones that are in black are prickled wild relatives of the ones that are in red that have lost prickles. So the red species are ones where prickles have been lost. And what we see in this group, which is again a small group of angiosperms, even the whole circular phylogeny in the middle isn't all of angiosperms. It's only, this is basically Solanaceae. Solanum is one genus within Solanaceae, but there's been lots of loss of prickles within the Solanium, and this is where this is the group of species that for part of this paper they focused on. What we see is that prickles have been lost 31 times within this group, and that was estimated using parsimony analysis. And I just want to point out again for my students that parsimony is the simplest scientific explanation and how that's the most likely answer. The fewest number of character state transitions is another way of defining parsimony, if you want to. And secondly, you know, the phylogeny is a diagram of the most parsimonious branching relationship between species in a group, and that branching relationship would have been arrived at using a parsimony of looking at the number of shared derived character states and the fewest number of character state transitions that was necessary to produce that particular phylogeny. And so this is a well-supported phylogeny of this group. I just want to point out that prickles emerged in this group about 6 million years ago, which is not that long ago. It's literally not that long ago in terms of all plant evolution. Angiosperms in general originated around 150 million years ago, and we see prickles must have emerged in this group around 6 million years ago, but then they've been lost 31 times. And we're going to use that loss as a way of getting towards the genes that led to the formation of prickles. So the simple story is that mapping revealed that this locus called prickles was the genetic basis at the beginning. It's a trait that was evolving in a Mendelian fashion, and even Edward East might have been able to discover that there was this locus, and you could call it big A, and that's prickled, and little a would be prickles, and they called it the prickles locus, and that's where people left it for a long time until this paper came along that did more work to try to understand what is the gene in that prickles locus. And as it turns out, at the beginning, the simple story is that it's a mutation in a cytokine and biosynthetic gene called LOG, and that mutation happens to be a splice site mutation, a mutation at the junction between two exons in this particular gene called lonely guy. And lonely guy is involved in cytokine and biosynthesis, and the story kind of makes sense because cytokinin is an important plant hormone involved in growth, and we're not going to go into too much more detail on the function of cytokinin, but finding this mutation is the key here. I want to talk just briefly about lonely guy. This is the paper that describes this mutant, lonely guy, written in lowercase when we talk about the mutant. This is a picture from that paper, and I show the paper in the upper left here if you want to go find it. On the left-hand side is a rice plant that is wild type, and it has a number of stamens and it has a pistil. The PI is the pistil. That's the female part of the flower. The lonely guy mutation has only one stamen and it has no pistil. So this is why they named it lonely guy. At first, when I saw this gene, I thought, oh, poor guy, poor molecular biologist. He's just so depressed with his molecular biology work that he named the gene lonely guy. And I just had this depressing image in my mind. But it turns out not to be that. It's very well describes the phenotype where you have only one stamen, which is the male part producing pollen, and there's no pistil for those pollen to engage in sexual reproduction with. So this is the lonely guy mutant. That's how this gene got its name. It just was kind of a coincidence, I guess you could say. Well, at the beginning, it's a coincidence. You have no idea where the mutation is that causes pricklelessness or prickledness. And it turns out to be in this gene for a cytokinin biosynthesis from the study that was published in 2007. So a more complex story is that QTL analysis or fine-scale mapping was performed to locate the genetic basis of prickle loss to this log gene. And I'm going to spend quite a bit of time here on this figure 2A. It's a lot of information, I know. I try to layer in information piece by piece, and so I'm going to do that now. And ultimately, we're going to get down there to the bottom right-hand corner where we have a red arrow pointing to a log homolog. That's a homolog of the log gene, and that's the location of the mutation that causes pricklelessness. But here, we'll start more simply. We're going to map it using this prickleless eggplant. This eggplant is within that group of spiny solaniums. We see the domesticated eggplant, which not surprisingly doesn't have prickles, not very consumer friendly, I guess you could say. There's this wild relative that has prickles, and so they use those as the parental phenotypes and cross them. Pricklelessness is shown in black and red here. Prickled will be a black or white square. I got some animations out of order there, but I think it won't matter much. I'm going to cross those together just like we did, or I suggested in a QTL analysis. The genome of the prickleless variety, which is the domesticated eggplant, is going to be gray. The genome of the prickled wild relative is going to be in black. And just remember that the parental genotype is black. The parental prickled genotype is black. The parental prickleless genotype is gray, not red, but gray. The phenotype of the prickled is going to be an open circle or a black circle. It doesn't matter much. Pricklelessness is going to be in red. That'll be easy to follow. All right, it's going to be an open circle. I forgot to switch that blacked in square. We'll use this, looking at the chromosome. This is on chromosome 6. We know in pricklelessness, this one region of chromosome 6 must have the genes. And in the prickled wild relative, it's the black genome. We're just describing the whole chromosome 6 here. We're going to generate an F1 population and then breed those together to create an F2 or mapping population. So this F2, we're looking at 5 individuals here, and you see how the recombination has broke apart the pricklelessness, the gray genome, and the black genome, which is the prickle genome, and recombined it into different chromosome 6s. Only some of the regions that are associated with pricklelessness have prickles or don't have prickles. And we see the phenotype on the right-hand side where it's open circle if it's prickled, it's red if it's prickleless. So the ones that are prickleless and also have the prickleless genome reveal the areas of the genome that are most likely to contain the genes for pricklelessness. And so I'm just circling or squaring off that in a red box there on the bottom. That's the area to look further into to find the genes associated with pricklelessness. So I'm going to magnify this up to a larger piece. It's the same figure, same exact figure. Look for more markers that are in that region. Markers meaning single nucleotide polymorphisms that are there in the prickled and not there in the pricklelessness phenotypes. Find more molecular markers in this 3 million base pair stretch. Still quite a bit of DNA, but not the 1.5 or 5 billion base pair. I can't remember how many base pairs the genome of the eggplant is, but it's certainly 100 or 1,000 times bigger than this region. We're down to 3 million base pairs, which is a lot, but still getting closer. Remember that single nucleotide polymorphisms were present in one parent and not in the other. So we're using that dichotomy to narrow down which markers are more closely associated with the phenotype. So these are markers that they found in that 3 million base pair region, and then we score for the presence and absence of the markers and for the presence and absence of prickles, and we see that 5 markers were useful. See that red box, the new red box, the smaller one, and then at the bottom we have another red box where we have only the prickleless genome, and all of those individuals are prickleless. That is, they have a red box on the right-hand side. This is probably the most complicated part of this presentation. But if you can understand this, you can see how we're using breadcrumbs to try to find the location of the genes that are controlling prickles. In that area, they used previously published sequences of the Solanium genome to find 10 annotated genes. 10 annotated genes, but notice the window has declined considerably. We're almost less than half, a tenth of a megabase pair. So it's a much smaller window. And there's 10 genes, and some of them don't matter what they are because they turned out not to be associated with pricklelessness. Of course, they went through all of these F2s. I would assume that you go through the F2s and sequence a lot of them and find the ones that have unique mutations, especially mutations that are in the same place. And all of that led to, well, this is what we call fine scale mapping. All of that led to, well, we call it fine scale mapping, where we're sequencing all of the candidate genes in the prickleless F2s and finding mutations associated with the phenotype. And that led to this type of mutation, a splice site mutation in the lonely guide gene. And now we can ask the question whether the other losses of prickles in the solanium also involve mutations in the lonely guide gene, and it turns out that they did. And this is the conclusion in figure 3 of a complicated presentation of the mutations within the spiny solaniums with these 16 independent losses, all involving log mutations, log meaning lonely guide mutations that led to the loss of prickles. They're a different type, each one of them is a different mutation. Just like every human is slightly different, each loss of prickles involves a slightly different mutation, but 16 times it was in the same gene, lonely guy, and various types of mutations. On the right-hand table, You see frame shift mutations, there were 7 of those. Splice site mutations, there were 7 of those. Transposon insertion, that was 1. A deletion was 1. The mutation type is in different colors at the top. The indel frame shift is in orange gold. The splice site mutation is in red. Transposon insertion is the triangle. And deletion is the slashed purple bar. That's number 15. If you're looking at number 15, you see, oh, that's the species. Selenium melongena is the one that had a deletion, a large-scale deletion. It looks like it might have knocked out an entire exon. Other ones involve splice site mutations in that one exon next to the larger exon. 7, 8, 9, 10, 13, and 14 are all splice site mutations in that exon. You see the transposon insertion in number 6, the selenium torvum. This is a really cool map that shows lots of different mutations, but they're all in the lonely guy gene. There were 31 losses. 16 of them were in the log gene. So it begs the question, what about those other 15? And unfortunately, I don't have an answer for you there. I'm sure they were somewhere else in the genome, or maybe they were regulatory mutations, and I should have done my research, but mostly I'm focusing on this result right now. So I'm going to go back to that backstory. That remember that Pruitt said in highly influential paper that adaptive changes in functional properties of hemoglobin are typically attributable to a few replacements at key positions. I also want you to remember the search for homologous genes is quite futile except in very close relatives. That's what Ernst Mayr said, and also that there are many roads that lead to Rome is true in evolution as in daily affairs. And so I'm just like, you're in this word salad, word field, like what's true? What is, give me the answer, what's going to be on the test type of thing. And the answer is both, I would say. That there were, for prickles, the mutations in the same gene caused the loss of prickles, but they were all different mutations. I don't think we saw a single case where it was the same mutation repeated. That would be remarkable. We're going to see a few cases in our next paper where it was the same mutation, but that was very, very rare. If we go back further, we see that log mutations were found to be associated with prickle loss across other species, across the entire vascular plant phylogeny. And so this continues to support the idea that lonely guy mutations are largely the epicenter for prickle loss, or you could flip that on its head and say for prickle formation. But then they use CRISPR to introduce these types of mutations in prickled wild seleniums, and they were able to re-engineer pricklelessness, which is a really cool modern follow-up study, I guess you could say. And I'm going to show you the results here, and they're just really cool. So they take a they remove pricklelessness in new selenium genome editing system, CRISPR-Cas9, you know the deal. We've got all these different mutations. I'm just giving you a little emoji there of the mutations. You just pick one of those mutations and put it into Cas9 and see what happens in that wild relative. It's easy for us to do here, sitting in our armchairs talking about it, but that's essentially what they did. I think they used multiple alleles. How they chose them, I don't know, but it's cool. You'll see the results, and they did it more than once. They did it 3 times, if not more, to remove prickles in many different species, including the desert raisin, which is a forage crop, as you'll see. This is what the desert raisin looks like. It has spines on the fruit. It has spines on the stem. It's a pain to harvest, but it's useful for animals, and I think even for Aboriginal people. They would eat this what they call desert raisin, but it's actually a tomato. They use the gene editing, and it produces this variety. No spines. just think if you're an Aboriginal forager, what a great advance that would be. Your farm species are going to prefer that fruit. Even you, if you're eating the fruit, are going to prefer that. So this could be a big advance. It's not really what the purpose of this research. Well, at least this part, I'm sure they really did set out to do this, but they became collaborators in this larger project. Um, but I don't think this is what Lipman and Jack Shatterley set out to do. But it's a great outcome. Bunch of issues there we're not going to go into whether or not GMOs like this should be done, but I'm just going to leave that there. Cool result though, incredible. Showing some more wild type and CRISPR on the right hand side. This is another species of Solanum, the Prenophyllum. Has prickles on the corolla on the left, you can see, and doesn't on the CRISPR edited. CR stands for CRISPR edited, I assume. And then another species, same deal. They just showed it over and over again that they were able to re-engineer pricklelessness, which is pretty cool, I think. Remember, I'm just going to review some glossary notes. Single nucleotide polymorphisms. Splice site mutation is a mutation where enzymes are joined together during mRNA processing. Parsimony is where the simplest answer is the best, or the most likely one, and it's used to reconstruct phylogenies, and then also to map character states onto phylogenies. Pleiotropy is when a gene or mutation has multiple phenotypic effects, and they were really lucky in this case where pricklelessness, the gene, the mutations that they introduced didn't have pleiotropic effects. The log homolog is an orthologous copy of log that seems to function solely in prickle formation without pleiotropy. CRISPR, again, a technique for targeted transformation, which may reveal pleiotropy, or it could. Here it did not. And fine scale mapping is various techniques like QTL and lots of sequencing to get closer to the gene, or maybe just simply finding more markers in a interval where you've identified where through other QTLs analyses that the gene must be. So we get back to our take-home points. I just like to read these again, where prickles have convergently evolved and been lost many times in plants in general, and the extent to which convergent phenotypes rely on shared molecular traits, or which are genes or regulatory regions, is hotly debated. Here the authors show that convergent losses of prickles share a similar but different mutations in a cytokinin gene called LOG, or lonely guy. And QTL and fine scale mapping were used to find the genes, and CRISPR was used to re-engineer pricklelessness in prickled wild relatives, just to kind of seal the deal. And the authors picked a simple non-pleiotropic trait. And I just want to point out that it's not always this easy, because even if Zachary Lipman had started with pumpkins, that is obviously a continuously varying trait, and I doubt it would be as simple as it is in dogs, where FGF is kind of a very discrete trait that controls that. I wanted to give you a few more bonus takeaway points from this study. QTL and fine scale mapping are ways of finding the location of genes or alleles that contribute to the genetic architecture of a phenotype. I'd encourage you to use this phrase, although I was a little bit disquieted when I read in Evolutionary Analysis, Evolution Evolving, new book by Scott Gilbert and Marcus Feldman and other people, that they don't like the word genetic architecture as much. I think they're very non-deterministic. They are trying to bring the environment more into the debate, but that's a different story. Sequencing genomes only gets you so far. A lot of my students are like, just sequence it, just sequence it. QTL analysis is a cleverly designed experiment that is going to save lots of time to get towards the genes that are controlling a trait. Remember, the genome is huge. It's like trying to find a needle in a haystack. Getting a 3D image of the haystack is not going to be that easy in finding that needle. Instead, doing something like a QTL analysis will be more useful. I think that analogy kind of breaks down there. Genome-wide association studies is another popular thing to try to do, but I'd say it's a modern shortcut that omits the cleverly designed experiment, and I say at peril. And in the end, what you get is just a correlation, and you don't really have kind of the smoking, the evidence just isn't quite as strong as it is from a QTL analysis. You have the phenotypes. You have the phenotypes. In a QTL analysis, you have the phenotypes and you have this association with the markers, and it's much stronger. CRISPR, if possible, can reveal and seal the deal for a mechanistic hypothesis from QTL studies. If you can, do that. And I'm sure Science and Nature and PNAS editors are asking everybody to do CRISPR to confirm that the QTLs that they've discovered actually control the trait. This must be kind of frustrating for people to get that comment back, but then when they actually do it, it's like, wow, that's amazing. They did show it. But if the QTL is spread throughout the entire genome, as it often is, CRISPR would be much more difficult. And that's about as you may not be able to do it, unfortunately. All right, well, that's the end of this presentation. I think in conclusion that Max Pruitt's was right, that a few replacements at key positions is often controlling traits, especially traits that are non-pleiotropic. I do worry that a lot of the examples that we show in classes and maybe even in publications will trend towards the simple side because they tell a more complete and satisfying story. I have a feeling that a lot of the traits out there are much more pleiotropic, and the answer may not be as simple as what Max Pruitt's suggested, and maybe even what is the conclusion of this paper. But I do not think that the search for homologous genes is futile, except for in very close relatives. I think that is the wrong statement coming from Ernst Mayr. And also this statement, though, that there are many roads that lead to Rome, is as true as in evolution as in daily affairs. I think I support this a little bit more, especially if you consider that in the log gene, there were many different mutations that led to prickle loss. So if you consider those different roads that lead to Rome, I think Ernst Mayr was probably fairly right here, even if he didn't say it. All right, take care.